Next, we're going to talk about some of the important properties that neurons possess. These include excitability, signal transduction, and conductivity. When we talk about excitability, we're talking about the ability of a neuron to respond to an external stimulus, a stimulus that's either coming from the outside world or from inside the body itself. Neurons can respond to stimuli. In fact, all cells in the human body can respond to stimuli. So this is a property that neurons share with other cells. Next comes signal transduction. Signal transduction is a process where a signal of one type is received by a cell and converted into a signal of another type. Like excitability, all cells have the ability to perform signal transduction. It's just that neurons are able to receive and convert signals into something pretty unique, which is electrical current. Neurons can receive an electrical signal in the form of what we call an action potential, and they can convert it into a chemical signal, something like the release of calcium or a neurotransmitter. Neurons are also able to receive a chemical signal, perhaps again a neurotransmitter, and convert it into an electrical signal. Neurons are capable of using signal transduction to communicate with other neurons, to communicate with muscle fibers, and to communicate with glands, both exocrine and endocrine. Last on the list of properties of neurons is conductivity. Now, unlike the other two properties, conductivity is quite unique. It is not shared by all cells. In fact, there are only two types of cells that have the property of conductivity. One of them is neurons, and the other is muscle fibers. Conductivity refers to the ability to generate an electrical current along the length of a cell's membrane. Cells that are capable of conductivity, again, neurons and muscle fibers, are able to conduct electrical current in the form of what we call action potentials. Now, we've talked about the difference between a single neuron with a single axon and a nerve. Remember, a nerve is a group of axons from several different neurons that are bundled together to transmit information in the body in the form of electrical signals. The speed of conduction, the speed of that electrical signal traveling along a nerve is dependent upon two key factors. One is the diameter of the nerve, and the other is whether or not it's myelinated. The axons inside the nerve are myelinated. Nerve signal conduction, in other words, occurs faster or slower depending upon these two things. Nerves that have a larger diameter are going to conduct electrical signal more rapidly. And that's because any nerve that has a larger diameter has more axons in it and therefore more membrane surface area. Remember, the nerve signal that gets conducted is actually being sent along the membrane of all the axons that are in the nerve. So this is a signal being transmitted along the membrane of each individual axon in each individual neuron. Myelination also plays an important role in the speed of nerve conduction. Myelination provides insulation. It allows an electrical signal to travel without any loss of strength because of the myelin that is helping to hold it in. So myelinated nerves, because they're insulated, are going to conduct more rapidly. Remember, it's not the nerve that is myelinated, it's each axon within the nerve that is myelinated. So we know that neurons have the property of conductivity, and we know how conductivity changes depending upon the diameter and the myelination status of a nerve. What we need to talk about now is how in the world neurons and muscle fibers are capable of generating electrical current. And it all begins with something called membrane potential. Now, all membrane potential is a difference in ion concentrations between the inside and the outside of a cell. Remember, we talked about ion gradients and how a gradient is simply a difference in concentration of something across a membrane. An ion gradient is just a difference in concentration of an ion inside and outside a cell. Membrane potential is simply a summation of various ion gradients that exist across the membrane of a cell. It turns out that all cells have membrane potential. All cells, as they exist in an organism, have certain ion gradients that are in place all the time, that are maintained across the membrane of the cell. When we examine all living cells at rest, there are certain key ion gradients that are the biggest players in generating this membrane potential. The key ions include sodium, potassium, chloride, and proteins. Now, when we talk about proteins, we're talking about the fact that there are many more proteins inside of a cell than there are outside. And proteins have a net negative charge. So the sodium is contributing positive charge, the potassium is contributing positive charge, the chloride is contributing negative charge, and the proteins are contributing negative charge. What happens with membrane potential is all of this charge and all of the different locations of this charge outside and inside the cell gets summed up, and it creates this resting difference in charge between the inside and the outside of a cell. An ion is simply a charged atom, and therefore an ion gradient is also a charge gradient, a difference in positive and negative charge between the inside and the outside of a cell. It turns out that in all cells, the resting membrane potential is negative, and it's negative to about 60 millivolts. You can actually measure membrane potential using a voltage meter. And again, it comes in at approximately, it varies a little bit from cell type to cell type, but approximately minus 60 millivolts. Now, what we mean when we say minus 60 is that the inside of the cell is relatively more negative than the outside of the cell. And it comes in as a sum of about minus 60 millivolts, inside to outside. On this slide, we can see a diagram of how those four key players of resting membrane potential, how they're distributed across the membrane of a cell. Remember, all cells have this resting membrane potential. So on this slide, we're looking at a little piece of plasma membrane here in the center of the slide. We're looking at the outside of the cell in blue and the inside of the cell in this brownish beige color. Now, the four key players are sodium, potassium, chloride, and protein. The sodium is represented by green circles. You can see those here and one here. Normally, sodium concentration is maintained higher outside and lower inside the cell. So there's a sodium gradient present at all times, with the concentration of sodium higher outside compared to inside. The second one is potassium. You can see the potassium in these brown triangles. Potassium is maintained at a concentration that is higher inside the cell compared to the outside, opposite of sodium. Chloride ion is shown in the yellow circles. Chloride is maintained higher outside compared to inside. And finally, there's protein. Protein is higher inside of a cell than it is outside. Now, there's a couple other things shown in this picture, primarily phosphate. Don't worry about the phosphate. That's not a big player in generating membrane potential. So, the sodium and the chloride are the two things that are higher outside. Sodium has a positive charge, chloride has a negative charge. The potassium and the protein are higher inside. Potassium has a positive charge, protein has a negative charge. When you add up all the positives and negatives outside and all the positives and negatives inside, what ends up happening is you have a charge gradient with more negative inside and more positive outside. And again, when you measure this with a voltage meter, the inside of the cell comes in at about minus 60 millivolts. That is resting membrane potential. That is a feature that all cells share, including neurons. Now, we've said in here before that charged things like ions are not able to get across a cell membrane very easily. We talked about how having a charge is one of the things that prevents a particle, a molecule, from getting across a membrane using simple diffusion. Remember, we said in order for ions to cross a membrane, they have to travel through proteins called ion channels. We said that ion channels are quite specific and they're not open all the time. Something has to happen to the ion channel to cause it to open. So there are channels in membranes that allow sodium to pass across the membrane. There are also channels for potassium. There are channels for chloride. Each one of these channels is specific for that one ion, and something has to happen to make that channel open. Most of the time, the channels are kept closed. During normal conditions in a living cell, a closed ion channel doesn't let sodium or chloride or potassium or any other ion go through. But ion channels are not perfect. They're not 100% perfect at preventing ions from passing through when they're closed. They are imperfect, just like all biological systems are imperfect. And so some degree of leakage occurs through ion channels. Again, most of the time, there's more sodium outside than there is inside. But the sodium channels that exist in this membrane are a little bit leaky. So sodium sometimes will move into the cell, down its concentration gradient. Leakage tends to be limited and it tends to be slow, but it's problematic because if leakage were allowed to just go on all the time, slowly, what would happen is more and more sodium would pass from the outside of the cell to the inside of the cell, down its concentration gradient. And if that were allowed to occur, membrane potential would be lost. This difference in charge from the inside to the outside of the cell would be lost. If potassium channels were allowed to leak uncontrolled, potassium would travel from the inside of the cell to the outside, down its gradient. Chloride would travel from the outside of the cell to the inside, down its gradient. There is no channel for protein, so we'll keep protein out of this discussion. So ion channel leakage exists, and if it were allowed to continue in a cell uncontrolled, it would be very problematic because it would eliminate the charge gradient that exists, and it would therefore eliminate membrane potential. So how does a cell manage ion channel leakage? The answer can be seen in this diagram. In this picture, we can see a piece of membrane across the middle of the slide. The outside of the cell is represented in yellow, the inside is represented in blue. All these little colored circles represent sodium or potassium. And these brown structures inserted in the membrane, those represent proteins. This protein on the right-hand side represents a potassium channel. This is an ion channel, and it only allows potassium to pass through. When this ion channel is open, potassium will travel through it down its concentration gradient. When the channel is closed, potassium doesn't go into the channel, except a very small amount of leakage. Now this structure over here, this is another protein, but this is not an ion channel protein. This is a pump. And this happens to be a sodium-potassium pump, or what is more correctly called the sodium-potassium ATPase pump. This is a pump that works by using the energy of ATP, by hydrolyzing ATP. It works to move ions up their concentration gradient. In other words, the sodium-potassium ATPase pump is what handles ion channel leakage and makes sure that it doesn't get out of control. This pump is going to pump 3 sodiums out, that's the little 3 purple dots, while it moves 2 potassiums in, that's the 2 yellow dots. Remember, normally sodium is higher outside the cell. You can see a lot more purple dots outside than you can see inside. Normally there's a gradient of sodium, and there's more sodium out than in. This pump is maintaining that gradient. It's constantly pumping sodium out. It's constantly pumping it up its gradient towards the side where there's already a lot of it. Potassium's the opposite. Potassium is normally higher inside a cell. This pump is adding more potassium to the inside of the cell. It's moving potassium up its concentration gradient. The pump is functioning this way in order to handle ion leakage. As sodium tries to leak into the cell and potassium tries to leak out of the cell, the pump is reversing that and keeping these gradients in place. This makes sure that all cells, including neurons, have their resting membrane potential intact at all times. And remember, that comes in at about minus 60 millivolts. For most cells, that resting membrane potential is going to stay at minus 60 millivolts all the time. But neurons are going to do something with it. Neurons have the property of conductivity. They're going to be able to generate electrical current from the membrane potential. So, ion channels are facilitated diffusion. Things move down them, down their gradient, from the side where the ion is higher towards the side where the ion is lower. Protein pumps move things up their gradient using active transport. Protein pumps require a source of energy to do this work. It's the pumps that maintain the different concentrations of ions across cell membranes. It's the pumps that maintain the gradients. The charge gradient that exists across all cell membranes is what we refer to as resting membrane potential, minus 60 millivolts. It's neurons that are able to use that membrane potential. They are able to allow ions to literally flow along the length of their membrane. And we're going to talk about how they do this. Another way to think of this is that neurons are able to take their membrane potential, that charge gradient, and put it into motion. When they do that, when they take that charge gradient and they put it into motion, what they are doing is generating electrical current. When we talk about nerve impulses, when we talk about nerve signals or electrical signals, we are literally talking about electrical current. And it is traveling along the length of the membrane of neurons. Remember, all cells have resting membrane potential, but only neurons and muscle fibers have the ability to generate electrical current. One other note here. The sodium-potassium ATPase pump is incredibly widely distributed. All cells have the sodium-potassium ATPase pump in their membrane, so it's very, very abundant throughout the body. If you look into the nervous system specifically, the sodium-potassium ATPase pump accounts for 70% of all of the ATP that gets eaten up by the nervous system. 70% of the ATP that gets consumed by nervous tissue in the nervous system every day gets consumed by the sodium-potassium ATPase pump. That's how abundant it is, and that's how important it is. And because cells generate ATP by using glucose and oxygen, it is the sodium-potassium ATPase pump that accounts for the fact that nervous tissue has an enormous requirement for glucose and for oxygen in order to make all that ATP that pump is going to eat every single day.